Good morning, grade 9 students. In this video, we're going to talk about our lesson about the mitosis and meiosis. So let's start with our PowerPoint presentation. To start with, we're going to talk about mitosis and meiosis. Mitosis and meiosis, they are the mechanisms of sexual reproduction. So I'm going to move step by step. First, I'm going to start with the mitosis process. Before, you have to know a small information that biology is the only science where multiplication and division, they mean the same thing. They mean increase in number. So mitosis is the process by which the somatic, so pay attention, this is the difference between mitosis and meiosis. Mitosis is the process by which the somatic cells of the body, they reproduce, meaning that the diploid cells, they produce new daughter cells. These daughter cells, they are identical, they are similar, they are the same as the mother cell, and they conserve the genetic information. Why would the body cells need to reproduce? These notes, you have to know them, to increase the cellular number and allow the growth of individual, and also to renew the body tissues and replace the old, dead, or the injured one. So you have to know that the cell undergoes interphase and mitosis. Interphase said that it had 3 checkpoints: G1, gap 1, S, synthesis, during which the replication or the duplication of the DNA took place, and we have G2, gap 2. And we have the mitosis, as I said before, prophase, metaphase, anaphase, and telophase. So here are the process. As I said, during the S phase, the sensitive phase, the DNA is replicated. So we have replication of the DNA. Cells as interphase, they are in the chromatin. We don't have chromosome, we have chromatin. So during the prophase, the nuclear membrane disintegrates. You have to study these events. The mitotic spindle fibers, they are formed. The centrosomes will change into asters and they move to the opposite pole of the cell. The chromatin, they condense, and that individual chromosome, they are distinguished. Each chromosome is composed of 2 chromatids, 2 sister chromatids. At metaphase, it's clear, they line at the middle of the cell, forming what's known by equatorial plate. During anaphase, sister chromatids and the spindle fibers pull the sister chromatids, they move the sister chromatids apart. Each chromatid moves to the opposite pole as shown here. Each chromatid moves to the opposite pole of the cell. This is what's known by polar ascension. Here, by the late anaphase, we have 2 identical groups of chromosomes. Each is made up of 1 chromatid. They move to the opposite pole. So what is special by anaphase is the equatorial plate. What is special by metaphase is lining at the middle of the cell, forming what's known by the equatorial plate. And we have during anaphase, the polar ascension. And last phase of mitosis, we have telophase and cytokinesis. Telophase and cytokinesis, the chromosome, they form 2 identical groups at the opposite pole of the cell. These chromosomes will decondense. They will come back to their origin, to their origin, which is the chromatin. Nuclear membrane reappears or reform. The spindle fibers, they disintegrate, they disappear, and the asters, they move to their origin, which is the centrosome. Cell membrane constrict. So this constriction is called cleavage, furrow, to divide into 2 sister chromatids. Okay, to divide into 2 sister identical cells. And you have to keep in mind that during the first node, the process where the cytoplasm starts to constrict, we call it the cytokinesis, where the cell is split into 2 identical daughter cells. And at any stage during mitosis, the cell has 2 end chromosomes. The 2 daughter cells they obtain after each cycle, they are identical. That's why the genetic information is conserved. They have the same genetic information. This is some notes about mitosis. And we have to move now to these figures during prophase, metaphase, anaphase, and telophase. Notes in plant cell, there is no asterisk. So this is an important note that you can distinguish between the plant cell and the animal cell. In the plant cell, there is no asterisk, while the animal, we have asterisk. No constriction at the end of telophase. Why? Because we have rigid cellulose wall, so there is no constriction. and we have new cellulose will form around the plant cell. And interphase, we said about interphase. Now let's move to gametes, specialized cells with 23 chromosomes. So here, move to meiosis. Meiosis is the process by which the mother cell of gamete, the mother cell of the gamete will produce the gametes. Gamete, they are haploid, they are 1N. So before meiosis begins, the mother cell will go through interphase to prepare itself to meiosis. Meiosis, we have two divisions, meiosis 1, meiosis 2. Meiosis 1 is called reductional division, and we will know why it's called reductional. And meiosis 2 is called equational, and also we will know the cause why it's called equational. During a prophase 1, as you see here, the chromosomes, they are lining up. Their sister chromatids, they move one over the other. They have pairing of homologous chromosomes, Fatima. So the homologous chromosomes, they pair up. Pairing up, they form what's known by tetrads. So here we have something called pairing up, is called crossing over. So they move to each other. Look here, we have one blue, one red. So we have crossing over, we have pairing up. So there is a piece of the red come to the blue, and one piece of the blue come to the red. This is called the pairing up or the crossing over. So prophase 1, and pay attention, meiosis 1, you have to write the 1 in Roman number. You are not allowed to write as a number 1. So nuclear membrane disappear, the spindle fibers, they start to form, the centrosomes, they change into esters, the chromatin condense, so that the individual chromosomes are each formed of 2 chromatids. So you see here, 1, 2, 3, 4 points, they are the same as mitosis. Here we have the difference. The homologous chromosomes, they pair up. Homologous chromosomes pair up, what's known by synapse, forming what's known by tetrads. Tetrads mean that we have 4. This will increase the chance of crossing over between the non-sister chromatids of the third homologous chromosome. This is the non-sister chromatids. Okay? Homologous chromosome, they are not sister chromatids. Here we have pairing up. That's what's the point of forming of the tetrads. So here, during the metaphase 1. Metaphase 1, this is the pairing of homologous pair on metaphase plate. So metaphase 1, the tetrads. So first, we talk about prophase 1, then moving to the metaphase. Metaphase, as we said before, the chromosome, they line up at the middle of the cell. But here, we don't have chromosomes. We have something special for meiosis. It's called tetrads. So tetrads, they line up at the equator of the cell, of the plate, of the cell, forming what's known by equatorial plate. So here, another difference. Tetrads are not chromosome, they line up. So this is metaphase 1. Moving to anaphase 1. Anaphase 1, we have polar ascension for sure. But we have here, we don't have the spindle fibers, we're not pulling the sister chromatids. This is another difference from mitosis. Here in meiosis 1, anaphase 1, the spindle fibers will pull the homologous chromosomes. So the homologous chromosome will be pulled of each tetrad apart. Here, homologous chromosomes are pulled apart, not the sister chromatids. So here, spindle fibers pull the homologous chromosomes of each tetrad apart. As I said, each chromosome will move to the opposite pole, forming what's known by polar ascension. Late anaphase, we have 2 groups of chromosomes. Each are made up of 2 chromatids at opposite pole of the cell. Moving for anaphase. Finish for anaphase, and then we move to telophase. Telophase, during telophase, the cell membrane will constrict, forming what's known by cleavage. For all of them, the same events, but they differ in 1 or 2 events. So the 2 daughter cells, they each carry 1 copy of each chromosome. Each is made up of 2 chromatids. So the obtained daughter cells, they are haploid. Starting with mother cell of gamete, it was diploid. Here we have haploid, 1N. So the number of chromosomes here in each cell is reduced. So here, why we say that meiosis 1 is called the reductional division. Why? because the number of chromosomes is reduced into half. So it's called reductional division. So the daughter cells, they are different from each other, and also they are different from the mother cell. This is for the telophase 1. The telophase 1, the chromosomes form two groups at the opposite pole of the cell. The spindle fiber, they disintegrate, they disappear, and the aster return back to the centrosome state. Nuclear membrane reform or reappear. Here we have to know an important note. Before we started the meiosis, we have interphase to prepare the cell to enter the meiosis. But between meiosis 1 and meiosis 2, there is no interface. So between meiosis 1 and meiosis 2, there is no interface. So whenever the cell started with interphase, prophase 1, metaphase 1, anaphase 1, telophase 1, it finished this process, it finished meiosis 1, then it started meiosis 2. In meiosis 2, we have prophase 2. But you have to know that here in prophase 2, we don't have one cell, we have two cells here. So prophase 2, we have two cells. So the mitotic spindle fibers start to form. Centrosome is changed into asters. Nuclear membrane disintegrates. Wait a minute. This process or these steps, they are similar to that in mitosis. Prophase 2 is similar to prophase in mitosis. So let's move. Metaphase 2, in the cells, in each cells we have two cells. The chromosome, they line in the middle of the cell. So chromosomes, they line up at the equatorial plate and the equator of the cell. So here we have telophase 2, metaphase 2, sorry, is similar to metaphase in mitosis. Here we don't have tetrads, we have chromosomes, they line up. And in mitosis, we have chromosomes line up at the equatorial plate. Moving forward, anaphase 2. Anaphase 2, as you see here, the spindle fiber pull the sister chromatids to opposite pole of the cell, forming what's known bipolar ascension. This is similar for the mitosis. So here, spindle fibers, they pull the sister chromatids of each chromosome apart. Each chromatid moves to the opposite pole of the cell, forming what's known bipolar ascension. By the late anaphase, the end of the anaphase, we have two groups, each made of one chromatid of the opposite pole. Until now, prophase 2, metaphase 2, anaphase 2, they are identical to the mitosis, similar. So anaphase 2, we finished. Then we have telophase 2 and cytokinesis. Telophase 2 and cytokinesis, every daughter cell is going to divide into two. We have two daughter cells. The result will be 4 daughter cells. So the chromosome form two groups at the opposite pole, and the chromosomes will decondense and go back to the chromatin. The nuclear membrane start to form around each set, and the spindle fibers, they disintegrate and the aster return to the centrosome. So the whole meiosis 2 is similar and identical to mitosis. There is no difference. So here we have the interphase and prophase 1, metaphase 1, anaphase 1. Prophase 1 forming tetrahomologous chromosome, they pair up forming tetrads. They line up at the middle of the cell, forming the equatorial plate, where we have polar ascension, but the sister chromatid remain attached to the homologous chromosome. They move to the opposite pole. And then we have this comparison between mitosis and meiosis in the diploid cell. In mitosis interphase, we have prophase, metaphase chromosome, they line up at the middle of the cell. While meiosis interphase, then we have prophase 1, we have pairing of homologous chromosomes, forming what's known by tetrads. At the end, we have duplication and the amount increases, and the homologous pairs of duplicated chromosome, they line at the middle of the cell. We said that instead of saying homologous pair of duplicated chromosome, you can say simply tetrads. While here in mitosis, the chromosome, they line up at the middle of the cell. In meiosis 1, metaphase 1, we said that tetrads, they line up at the middle of the cell. Let's continue the process here. Anaphase in mitosis, the sister chromatids are pulled to the opposite pole, telophase, and the result will be two daughter cells, and they are identical to the mother cell. Here we have the telophase 1, we have cell division, we have metaphase 1, anaphase 1, telophase 1, and meiosis, metaphase 2, anaphase 2, telophase 2, and then we will have the final result. Every daughter cell is going to divide into two. So we have two daughter cells. So the result will be 4 daughter cells. This is the difference between mitosis and meiosis. So mitosis will give 2 daughter cells, whereas meiosis will give 4 daughter cells. And these figures are representing what I'm explaining about. So here, during meiosis, the chromosome number in each cell is reduced. During meiosis 1, it's reduced. That's why it's called reduction. Why? Meiosis 2, it's called equational. So it's going to be equal. All the daughter cells, they are having the same number. So here, 4 daughter cells contain 1 chromosome each. These cells will become the gametes. That's why meiosis 2 is called equation, because we have equal number of chromosomes in each cell. This is the same figure. In mitosis, we have 2 daughter cells at the end, while in meiosis, we have 4 daughter cells. So this table is very important. This table is very important for the comparison between mitosis and meiosis. You have to know that mitosis, it occurs in somatic cells. In all the cell except the sex cell, except the sperm and the ovum. So somatic cell, mitosis, while meiosis, it occurs in gonads. Mainly, the aim of mitosis is for the growth and renewal of replacement of the injured or dead cells, while the aim of meiosis is production of haploid gamete, sperm and ovum. One division only in mitosis. We don't have mitosis 1 and mitosis 2. So one division only, producing how many number of cells? 2 daughter cells. While in meiosis, we have 2 divisions, meiosis 1 and meiosis 2, and they are not separated by interphase, producing 4 gametes, 4 daughter cells. Mother cell and daughter cell, they have the same number of chromosomes. They are identical. Why? The number of chromosomes is reduced into half, coming from mother E2N to the gamete, which is 1N. Mitosis occurs to somatic cells in all of the body organs. So it occurs to the somatic cells. Why? Here it occurs to the mother cell of gametes and the gonads. It's made up of only 1 division, as we said. Here it's made up meiosis of 2 divisions. They are preceded by interphase. Here we don't have pairing of homologous chromosomes. We don't have synapse. We don't have the formation of tetrads during mitosis. While during meiosis, we have pairing of homologous chromosomes. We have formation of the tetrads. Daughter cells obtained are 2N, 1 chromatid per chromosome. They are diploid, 1 chromatid per chromosome. And after meiosis 1, daughter cells 1N, 2 chromatids. And after meiosis 2, the gametes obtain 1N, 1 chromatid per chromosome. So here you have to know that, as I said before, note that meiosis 2 is greatly similar in its events to mitosis. Similar, except at the end we have 4 daughter cells. In mitosis we have 2 daughter cells. In meiosis 2, prophase 2, metaphase 2, anaphase 2, telophase 2, they are greatly similar in their events for mitosis. Here, the number of possible different gametes is equal n equal the number of possible different gametes. N small is the number of pairs of chromosomes. For example, we have 2 pairs of chromosomes. So number of possibilities 2 to the power 2, so it's going to be 4. Notes, we have 3 processes, 3 biological processes. Either we have mitosis. This process will lead to the growth and the displacement of the old or injured or dead cells. Meiosis lead to the formation of gametes. Fertilization, it restores the diploid state and it forms a zygote. So both meiosis and fertilization, they lead to the diversity and the differences in genetic information. Why? Because meiosis and fertilization will lead to different gametes, will lead finally to the differences in genetic information as well as in the diversity. By this, we finished the presentation for this video about the meiosis and mitosis. Hope this video will help you to understand very well the differences between mitosis and meiosis. Here